ahead and get started. So the way class is going to work today, hopefully you watched that pre-class lecture that I assigned. If you didn't, you might be totally lost in the second half of class. But the first half, we're going to do a short lecture, kind of recap a little bit of that pre-lecture that I had you watch, our pre-class lecture. And then the last half, we're going to go outside and practice cell membrane transport with a game. So I'm not going to actually talk too much today. Now I know we left off on our last lecture with just a few slides remaining. So our last lecture we were talking about the plasma membrane. Let me turn on our captions here. Let's go that. There we go. All right, so we talked about our plasma membrane, right? We talked about what they're made up of, how it's bilayer, right? Those two layers, they're asymmetrical, so they're not identical to one another. Made up of those phospholipids, but also with different proteins scattered about. Whether those proteins are crossing both layers, attached to one layer, or is a membrane that might be, or excuse me, a protein that might be attached to another protein that is embedded within the plasma membrane here. What we did not get to is the fluid part of our mosaic fluid model. So, membranes, they are not stagnant. They can move, all right? They're not going to stay the same format, the same arrangement the entire time, right? There's movement that can be had, right? A couple of different ways that movement can occur. The first type of movement is just rotational movement, where if we have a lipid in our membrane, we can just kind of rotate in place. Right, so rotate around. Pretty simple movement. The other type of movement that a phospholipid can do is simply moving back and forth. So within that one layer, that one bilayer of our plasma membrane, it can switch positions. It can move laterally. Right, so it can rotate in place. It can move laterally. And then there's one more type of movement that it can do. It can actually do what we call a flip-flop. So if we have our membrane here, our phospholipid can move from one side of the membrane to the other side of the membrane. Now this is less common because it is a little bit more complex. It takes energy and it needs an enzyme in order to mediate this movement. The reason being, right, if we remember the inside of our membrane here, this is a nonpolar region, versus the head of the phospholipid is polar. Right? So that polar head really does not want to travel through that nonpolar region. Right? So in order for it to do so, it needs to have this enzyme called flippase, right, aiding the flip-flop movement, as well as some energy to make it move. Right, so rotational movement, lateral movement, flip-flopping movement. Right? This is why we would say if membranes are semi-fluid. They are not rigid. All right, there are different factors that might affect how fluid a membrane is. And it all has to deal with basically the phospholipid here. So if we look at our phospholipid, we have our phospholipid head and then our two tails here. Right? Our tails are nonpolar, our head is polar. These tails can have different lengths. Right? So those fatty acid tails can be of different lengths. If they are shorter, right, they're not going to interact with the other side of the plasma membrane as much. Right? So they can make that membrane more fluid, meaning that phospholipid can kind of move around a little bit more easily. Now, if those tails are longer, right, now the tails might be touching, and to try to move around, there's a little bit more friction, right? It's going to get tangled up in the tails of the other side of the membrane. That would make it less fluid. So the length of those tails is a factor in how fluid this membrane is going to be. But also, if we remember saturated versus unsaturated fatty acids, right? So saturated has the maximum amount of hydrogens that could possibly be bonded to our fatty acid. Unsaturated typically has a presence of a double bond, right? So it doesn't have as many hydrogens. Well, that puts a bend, that puts a kink into the fatty acid tail, right? And when that happens, it also, again, makes it harder to interact with its neighbors because it kind of has that bend, right? You can see, I like this image up here, because you can see when it has this bend, there's a little bit more space between it and its neighbor. Right? So it's harder to interact, harder to get tangled up, essentially, making the membrane, once again, more fluid. Right? So not only the length of the fatty acid tails, but the shape, right? Is it saturated or unsaturated, is going to determine how fluid a membrane can be. The last thing that can affect fluidity is not actually the phospholipids themselves, but the presence of cholesterol that can be embedded within our phospholipids. So cholesterol, right, tends to stabilize membranes. And how it stabilizes membranes differs depending on temperature, right? So we see we have cholesterol here. It's kind of inserted into our phospholipid bilayer. It really depends on temperature. So if we look at a membrane that is in a cold environment, that membrane is going to be more rigid, right? Not as fluid, just because it's cold. Molecules tend to interact a little less, right? If we don't have cholesterol in there, right, that rigidity makes it a little bit more breakable, right? It's not going to bend, it's not going to flex as much, therefore it might actually break, right? Which, if our plasma membrane breaks, that cell is essentially going to die, right? Because everything on the inside is going to leak out. If we insert some cholesterol, right, it's going to make that membrane a little bit more fluid in colder temperatures so that it does not break. The opposite applies for when it's hot out, right? So when it's hot, a membrane can actually be too fluid, right? It moves around too much, again, making it unstable, making it too flexible, and it cannot hold the shape of the cell. So again, we're going to insert cholesterol to stabilize that membrane and make it a little bit more rigid, right? So cholesterol is stabilizing, and its effects depend on the temperature. Now we talked about our phospholipids, right, how they can move, talked about fluidity, but we know there's also proteins embedded within our phospholipid bilayer. Some of these proteins will also do the same types of movement, right? They can move laterally, they might be able to flip-flop, right? That's going to take a lot of energy though. But there are some proteins that just simply won't move because they're directly connected to the cytoskeleton or the extracellular matrix, right? So there's less movement of these larger molecules, less movement of these proteins, and they're not going to move at all if they're directly connected to the cytoskeleton or that extracellular matrix. All right, so our membranes, they are semi-fluid. There is flexibility in them. So I want us to review, why would we call this the fluid mosaic model? So I want you to define why it's considered fluid, and why it's considered a mosaic. I'm going to have to review notes from last week as well to get through this. I'll give you a few minutes. Don't test me on that. But I see. Yeah. That's very wise. That's kind of what I wanted to do. I wanted to leave you asleep just a little bit. All right, why would we call the plasma membrane or the phospholipid bilayer fluid mosaic model? Let's start with the first one. Why is it fluid? Yeah. There's movement. Yes, it's movement. It's not stagnant. Phospholipids can move. Proteins can move. What about mosaic? Why would we call it mosaic? Yeah, there's variety in it. Those two halves are not the same. They're a mosaic. They're a combination of phospholipids and different types of proteins, cholesterols in the mix as well. So the plasma membrane we would call this fluid mosaic model. Any questions there? All right, I had you watch that video on the types of transport because if I had to do that lecture with this lecture, we would have not had time for a game. But I did want to review it, right, just to make sure everyone's kind of on the same page here. So in that lecture, we went through the different types of transport. Either passive transport, which takes no energy, right, or active transport, which does require energy, right? And I'm just going to summarize it all in a couple sentences here, just to make sure if there's any questions, now would be the time to ask them. Right? Diffusion, we are moving molecules through the plasma membrane, through the phospholipids themselves, always going from an area of high concentration to an area of low concentration. That's what drives the movement. Right? That movement of high to low concentration is also the driver for facilitated diffusion. Right? But facilitated diffusion, we need to have this embedded protein. Right? Either the molecule is really large, it can't fit between those two layers of phospholipids, or the molecule is polar or has a charge, which means this hydrophobic interior is going to repel that molecule. Right? So it needs a protein to help it along. Then we talk about active transport. Right? Where this time the gradient is switched on us. We're now going from an area of low solute concentration to high. Right? This is not how the molecules want to move naturally. Right? So to get them to move across that gradient, against their gradient, we need to have that form of energy. Right? Energy is provided along with the protein here that moves the molecule across. And remember we talked about two different types. We talked about primary and secondary transport. Primary transport directly uses the ATP. Secondary transport, right, relies on primary transport to build up the gradient, and then it's that gradient that moves molecules across the membrane. Any questions from the video? All right, I'm gonna have us do just a quick review before we move on. All right, so thinking about bringing solutes across the plasma membrane, what are the advantages and disadvantages of those three transport types? All right, simple diffusion. What's the advantage of simple diffusion? Yeah, it doesn't need energy, doesn't need ATP. What might be the disadvantage though? Yeah, it can only move simple things, things without a charge or small molecules. Right? Not everything can just simply go through the phospholipid bilayer. What about facilitated diffusion? What's the advantage there? Yeah, again, there's no energy needed. That's a huge advantage. What's a disadvantage, though? You need that protein, so that means it can only go through the membrane at specific places, but that also means the cell has to synthesize that protein and embed it within the membrane itself. It's a little bit more complex, right? It takes some more cellular processes to make it work. Active transport, what do we know about its energy needs? It requires energy, and that's definitely a disadvantage, right? But what's the advantage to active transport, though? Yeah, it can go against a gradient. Molecules naturally want to go from areas of high concentration to low concentration. That way, essentially, they can equalize on both sides of the membrane. Right? So going against it, where we're making a low concentration even lower by losing those molecules, It's not what a molecule wants to do, so you kind of have to force it to do it. Right? You have to use that energy. But there are instances where the cell needs to do this. Any questions? All right. I wanted to touch on osmosis because I did not touch upon it on the video. Osmosis is a type of passive transport, so it does not need energy. It's a type of simple diffusion, so it doesn't need a protein embedded in the molecule. In osmosis, typically what we see is that molecules, the solutes, what's dissolved in the water, is not going to move, right? Maybe it can't move. Maybe there's no protein to aid its movement. Or maybe there's just some other barrier to its movement, right? But the water can move. Those water molecules, they can go through a plasma membrane, right? They can use simple diffusion. Right? So the water is moving instead of those solutes, right? So if we look at this example up here, right? We have this U tube here, right? We have a solute molecule, which are in purple. If we look at the concentration across this barrier here, we see on one side there's a lower concentration of those solutes, and the other side is a higher concentration. Right? Now the solutes can't move, but the water can. So the water is going to flow across this membrane into the other side because we want to reach this equilibrium. So you can kind of think of it as the ratio of solute molecules to water molecules is higher here. There's much more water than there is solutes. We want to equalize that ratio. So water is going to move to the other side, right? So on both sides of the membrane, the number of solutes to water is the same. Even though that might mean if we have more solutes, there's going to be more water. Right? So think about it as equalizing those ratios, right? Water to solute ratio needs to be the same on both sides, and water is going to move accordingly to make it so. Typically, yeah, more solutes, more water. Right? So we want to maintain that equilibrium. Right? And again, I have this second diagram here. We have this selectively permeable membrane. Right? The water is moving across it. We see in this instance the barrier is size. Those solute molecules are too big to fit across these proteins or these gaps. And we want to equalize the number of solutes to water ratio. So we're going to have many, many more on the right side to equalize that out. All right, so there's some terms associated with this water movement and how water might move, right? So if you look at the solute concentrations, right, we can always compare it to another solution. When we're comparing two solutions together, or two sides of a membrane, right, we can use this terminology on the screen. We can say that those solute concentrations are equal, which would be an isotonic solution. Right? Now, I couldn't say that this one solution is isotonic. That means absolutely nothing unless I compare it to another solution. So it always has to be in comparison to something else. So an isotonic solution, equal number essentially of solutes on either side of a membrane. Hypertonic solution means that the solution concentration is higher. So we're looking at these images here. The outside is what we're looking at, outside of the cell in these images. The outside of this middle one is hypertonic because there's more molecules, there's more solutes. So we would say it's hypertonic to the inside of the cell. And then we have hypotonic, right? Exactly the opposite. The number of solutes outside of this cell is lower than the number of solutes inside of that cell. Okay. Now, this terminology is important to understand because, again, this is going to dictate how water is going to move in order to equalize the water to solute ratios. We look at some examples of exactly why this is important. Let's look at red blood cells. Right? If they're in an isotonic solution, so inside and outside of the red blood cell, equal number of solutes essentially. If we put it in a hypertonic solution, meaning outside of the cell, there is more molecules. The water is going to want to leave the inside of the cell to the outside of the cell to try to equalize those solute to water ratios. So we get cells that will shrink. Okay? Opposite is true. If we place a red blood cell in a hypotonic solution, right, so a solution has fewer solute molecules on the outside compared to inside of our cell, the water from outside is going to want to rush inside of the cell, and it could actually burst those red blood cells. Same concepts with plant cells here, except we know that the cell wall maintains that rigidity, so we're not going to have cells bursting or shrinking, but we can see the plasma membrane will shrink up inside of the cell if it's in a hypertonic solution, so water's leaving the cell. And we can see that that membrane might butt up directly against that cell wall if it's placed in a hypotonic solution. Right? So water's entering the cell. Right? So that cell wall is kind of more stabilizing for those plant cells. We're not going to see cells bursting per se. All right. Why do we care about water balance? Well, it's crucial to many organisms and to regulate many different processes, right? We could talk about its tonicity, which is the ability of a solution to gain or lose water, right? Less concerned, you know, that word versus osmoregulation, which is just the regulation, the control of our water balance. Meaning we're making sure we don't have so much water entering into our cells that they burst, or vice versa, that they shrink too small. Right? So osmoregulation, we're controlling our water balance. Especially aquatic organisms, if they are living in water, right, they might take in a lot of excess water. Well, that excess water might not be great because it might cause their cells to swell, so they have to have ways to get rid of it. Now, water movement moves via simple diffusion, but it can also move via facilitated diffusion, right? So diffusion is great, right? But if we want a lot of water into a cell, say we're looking at the roots of plants, right? The roots of plants, they take up water from the soil. We want to take up massive amounts of water. So they're going to have these specialized channels called aquaporins. And they will use facilitated diffusion to essentially move the water across these membranes even faster than it could via simple diffusion, right? So it's a channel very specific to water. Greatly increases the rate of water movement. Also found in animal cells, like in our kidney cells, we're constantly regulating the solute and solute concentrations of our cells. So water can move via osmosis, simple diffusion, but certain cells, if they have high water requirements, will have aquaporins, which is part of facilitated diffusion. The water can move both ways, yeah, depending on the gradient. Yep. So in facilitated diffusion, it's always going to go from an area of high concentration to low. Depending on which one is high and which one is low is going to determine which way it moves through that channel. All right, let's practice this. So, say you graduated Gustavus, you went to medical school, you're a practicing doctor now. This doctor wants to inject a patient with an isotonic saline solution. But unfortunately, the patient died, right? And an autopsy revealed that several of those red blood cells had burst. Is it true that this doctor injected the patient with an isotonic solution? I'll give you a few minutes to think why it might be true or false and what the correct solution might be. All right, I feel like most of you have finished discussing, so I'm gonna cut us off there so we can get to the is this true or false? Did the doctor inject the patient with an isotonic solution? Did not. It is false. What type of solution did he inject? Hypotonic. Yeah. So if we look at a red blood cell here, right, if it's a hypotonic solution, which means there's fewer solutes in the solution compared to inside the red blood cell, water is going to rush in the red blood cell and then they can burst. All right. I wanted to briefly touch upon this, right? We're not going to go into any super complex detail here, but we talked about movement in and out of a cell, but what about movement between cells? Will cells have the ability to create these channels that will span two different cells, two different adjacent cells, to allow for direct transport of materials from one cell to another? So in animal cells, we call them gap junctions. So we can see here we have two membranes lined up against each other, and we have a channel through both of them. Right? So these would be our gap junctions. And then we also have our plasmodesmata, same concept, right, in our plant cells. Right? Our plant cells, it's actually the plasma membrane that's continuous. Right, so two cells lined up against each other, they will have ways of directly moving solutes back and forth so they don't have to leave a membrane and then enter into another membrane. They can just go directly across two membranes. Another way that we can get movement into and out of the cell and between cells are these two terms we call exocytosis and endocytosis. And these are used to transport really large molecules, molecules that might not move via gap junctions or plasmodesmata very well, they're just too big. In this instance, right, exocytosis is when we have large molecules leaving the cell. Endocytosis is when molecules are entering the cell, and they're doing so using parts of the plasma membrane. So to leave the cell, right, likely they're going to be in this vesicle. Maybe the Golgi apparatus has packaged something up for us and it needs to leave the cell. That membrane is going to combine with the plasma membrane of the cell itself, right, and thus release it to the outside of the cell. Opposite is true if we want to take in molecules. So these molecules will kind of come into this plasma membrane area. That plasma membrane is going to fold in on itself, right, and then be cut off from the rest of it to form this vesicle. And then it can be transported again to where it's needed. All right, I didn't want to spend too much time on this. I wanted to focus on membrane transport with molecules. before we move on to the game, I do want to mention that Content Check 2 is due in class on Monday, and I'll give you part 2 in class. Remember, you have that scientific literacy assignment coming due next Wednesday. Over the weekend might be a great time to do it. It's a little bit longer. It's not hard. You just have to watch a few videos, read a couple articles. And the exam next Friday. It's coming up, right? So start studying now. The last content for the exam will be delivered on Monday. All right. We're going to go outside and play a game because it's nice out, and I feel like having practice with membrane movement is important. This is a long version of the game, but essentially, once we get outside, we'll split into 2 groups. Some of you will be the cell membrane. Others will be molecules. All right, so some of you might be holding up a sheet that says, I am part of the cell membrane that can do simple diffusion. Right? If you have a molecule, you're going to have to come up to the cell membrane and ask questions about your molecule. You don't know what you are, right? Which is why these are folded in half. You're not going to look at your molecule. Instead, you're going to go up to somebody and you're going to open it so they can see it and say, oh, is my molecule polar? Or is my molecule large? Right, so there's some sample questions on this piece of paper to help you. It's the person who's a cell membrane. It's responsibility to tell you, are you a large molecule? Are you polar? When you feel like you know how this molecule is going to move, you'll go up to a person that's a cell membrane and say, does this molecule move via simple diffusion? And if they say yes, you can come on through. If you're part of the membrane, right, you do have the answers on the back to help you know this. All right, so let's head outside. We're going to head outside just outside these windows here. We are not going to come back to the classroom, but you can leave stuff in here if you'd like.